Hello everyone. Today I'm going to talk about biological networks. So what do we mean by systems biology? An approach to have better understanding about the larger picture at the level of the organism, tissue, or even a cell by putting its pieces together. So here we are talking about a bigger picture. Before we were trying to find, for example, alignment for genes, for proteins. We were trying to compare them in terms of sequences or structure. So everything was related to proteins or mRNA or genes. But now we are trying to understand what are the interaction between them. So we have so many proteins. What are the interactions? We have genes. What are the interactions with genes and proteins? So we are talking about the bigger picture. Why do we do that? Because we are trying to understand about the function of, for example, several proteins together to understand why they are responsible for a specific task in our body, in our organism. For example, we are even discussing about a specific tissue, how it happens, how we have this tissue, or how do we have this organism after all? What are the differences between different organisms and which proteins, for example, we have so that we have such an organism. So these things we can talk about in system biology. And we have a term gene regularity network, a collection of molecular regulators that interact with each other and with other substances in the cell to govern the gene expression levels of mRNA and proteins, which in turn determine the function of the cell. So you see, again, we are talking about the function. And also gene expression. We talked about it in our first lecture briefly. But when we say gene expression, it means that how much of the protein or mRNA is synthesized or produced by gene. For example, if we are talking about beta globin gene, so what would be the level of beta globin mRNA or beta globin chain as proteins? And we can talk about it through the gene expression. Whenever we can see that we have high level of mRNA or protein, then we can say that the gene expression is upregulated because the level is increasing. And whenever it is decreasing, we say that the gene expression is downregulated. So these two terms also are used for gene expression. So gene expression is very important, and whenever we talk about it, we usually emphasize on the levels of mRNA and proteins, okay? Because whenever you hear about gene, you think I should talk about the gene level. No, usually we are talking about the mRNA and protein levels. So in our simulation results, usually you will see the protein and mRNA level. So this is an example that how it looks like. So we have so many proteins, genes, and mRNA that we should find out about the connection between them, the interaction that they have. For example, whether a specific protein, for example, this one, whenever this one has, for example, the gene expression is upregulated, what happens, for example, to this protein? Whenever mRNA level is high for this one is also experiencing the same thing or not. So if we find out about the correlation between them, maybe we can find a connection between these two, because these two seems to be far away, but there might be a connection. And maybe the connection is not direct. Maybe it is through this one, for example. So there is a reason that whenever the mRNA level of this one is high, the gene expression of this one is upregulated. So these are the things that we should understand through such networks. But as you can see, this is complicated. We need now computational methods, we need mathematical modeling in order to deal with that. And that's why computer scientists could be very helpful for bioinformatics, because we need to transform everything into a model so that by using computer-based simulation, we can find out about some outcome that we couldn't see that like this. Assume that we know all the interaction, which is not the case, but assume that we know that, but still it's not easy to see what happens for each one of these proteins. So mathematical modeling can be very useful. But the picture that you saw before this was such a mess, right? So we need pathways. Pathways shows specifically a part of our biological network, which is responsible for a specific function. And if we can find out about such pathways, and molecular biologists were very helpful for that. Because of their experiments, they could find out so many pathways. Can we say it is 100%? Again, we cannot say that. Maybe there are some other factors involved in it that nobody knows until now. For example, maybe a specific protein is involved also in the same picture that you can see as in this pathway that nobody knows now. Maybe in the future, for example, they understand there is another protein, and this protein actually upregulates the gene expression of this one. So this one, this specific protein, for example, should be inside the pathway, but it is not included in this picture, as you can see. So scientists are helping each other through this to find out about the correct pathways, and that's what is happening. But these pathways, they are very useful, and there are databases which contain these pathways. I already introduced you to the best one, KEGG pathway database. So whenever you want to find out about a pathway, just you can look up for it here. You can search it and press go. There are so many of them, fortunately, and whenever personally I want to know which pathway is interesting, and for example, I'm assume that I'm working on brain tumors, then I can search for it, that whether I can find a pathway here related to that or not. And there are actually some of them here on KEGG as well, and that maybe initialized my research. So whenever I want to initialize my research in bioinformatics, I prefer to start it with a pathway, because these pathways can be interesting for me. Maybe there are things that are not predicted so far. For example, the role of this protein. Maybe nobody investigated what's going on exactly, the important role of this one. So in molecular biology, they are running these experiments on different species, because it's not ethical to run it on human being. This is the same situation that these days you are hearing about. That first they are running experiments on animals, then after that, if it is safe, they try to do it on some volunteers as human beings. Then after, for example, a few months, when they are sure that there are no side effects, then they say, okay, we can distribute the medicine to large population. Whether this is ethical to run it on animals is also another story. But when we see, for example, doing this will lead to saving so many lives, then it makes sense. But of course, it's on debate that whether it is also ethical to run it on animals or not. but this is what is happening. And molecular biologists are trying to find out about the role of each one of these proteins or genes or mRNA by, for example, upregulating or downregulating any of these components. So if, for example, let's say this one is downregulated, then they will see whether the function is still working properly or not. And why they are doing this? Because, for example, let's say in a human being, because of a mutation, this component is downregulated. And because of this multi-protein is not functional as it should be. So how can we compensate it? So by, for example, upregulating this component, maybe it's happening. Or, for example, the interaction it has with other molecules. All of them can be manipulated. But are they easy in terms of molecular biology? Is it, for example, cheap to do that for different experiments? Of course not. This is time consuming, this is costly, and we are talking about the lives of animals as well. So how can we avoid that as much as possible? They are actually very useful, and this is the direct way to do that. But how can we make predictions and see things that molecular biologists couldn't see before? So by using computer science, by using computational models, we can do that. We can predict things that say, okay, molecular biologists, don't do it for all of these proteins one by one. Let's find out which one of these components have the most important role as a prediction by simulation. I could see, for example, this one. Then if you are running tests, let's start with this one. And if we reach the result that we were looking for, then the experiment is a stop there. That's it. So we are not going to run more tests, for example, in this pathway. So, and also I can make some prediction like this protein maybe is important to be considered inside this pathway. That's why mathematical modelings and computer simulations are very helpful for bioinformaticians. And that's also another reason we have a major bioinformatics, so that those people who are interested in both computer science, mathematics, and biology can have them all in this major. So the thing is that when we are trying to go through these pathways, because that's what we have. When you search on KEGG, for example, you receive such pathways. So how can I model them? Next week, I will talk about it in details, that which models can be useful in order to do that. But what's the goal here? The goal is that to transform everything into a model like this one. This is actually a PetriNet model that I will talk about it next week. So then you will see that each one of them, for example, these are places, these ones are transitions, and each one of these arcs, they have weights, for example, and also these transitions, they could have a kinetic parameter. They could have rate as well. So to find out what's the rate, of course, it's time consuming and it is not 100% for sure. This is also our challenges to deal with that. These are our challenges to deal with that, so it's not easy. So we should read so many articles. We should get the help of molecular biologists so that we get idea, for example, what should be the rate here. But there are so many challenges that I will talk about them in details next week. And what's the goal here? We have a mathematical model. Let's say this is a mathematical model, PetriNet. We have a directed graph, as you can see. You already know about graphs in one of your courses, right? So that's why this is an application of a graph. As you can see, they are very useful. Instead of using so many derivatives, so many equations, now we transform everything in terms of graphs, which are visually even easier for us to deal with that. The goal is that, first of all, we try to describe the biological system. It means that here I have a pathway. Now I'm trying to find a mathematical model which is a good match with this one. If this is true, then by manipulating each one of these components, then I can analyze this biological network. And that's the beauty of that. It means that first I should validate whether this model is a good description of that biological network. If I can show that, then as soon as I manipulate the components, then I can have some prediction, which was very costly if I wanted to do that as a biologist, for example, in labs. So that's why computational methods can be very useful. So this is a big advantage of mathematical modeling or computational modeling. You may say, what's the disadvantage? The disadvantage is that by accident, we think that this is a good description of the biological network because everything is artificial here, right? This is not something natural. And maybe we are having some errors. By accident, we reach exactly the same, for example, mRNA level or protein level for all of the components. The gene expression totally match with the one that we have in nature. But in reality, when we are, for example, upregulating or downregulating a specific component, it doesn't happen in nature. So this is also a disadvantage. But what is the main point here? The main point is that we are saving time, saving money of molecular biologists here anyway, because in so many ways they were helpful, and the model that we have designed was actually the correct one. So if that was the case, so we are saving so many times, for example, for a disorder or disease that we could solve the problem for so many years, we solved, for example, in few years. So computational models and mathematical models can be very useful for describing biological networks. And next week, I'm going to talk about different mathematical methods or computational methods that we can use for describing and analyzing biological networks. Okay, guys, if you have any questions, please let me know. Otherwise, on Wednesday, we are going to talk about our last assignment, assignment number 10. There is also a formula correction, which I should talk about on Wednesday related to cosine formula. So please don't miss attending or watching that lecture.